Hello, everyone, and welcome to the MMID Coding Workshop series. Our instructor for today is Jessy Slota. Jessy is a graduate student in the Department of Medical Microbiology and Infectious Diseases at the University of Manitoba, supervised by Dr. Stephanie Booth. Today, Jessy will provide basic analysis of RNA-seq read count data, such as processing raw files, normalization, differential expression, and functional enrichment analysis in R using DSeq2 and enrichR packages. You'll also get to learn awesome RNA-seq analysis-specific data visualizations. All right, Jessy, the floor is yours.
Okay, thank you, Grace. I guess I'll just jump right into it. Okay, so yeah, there's basically 5 different things we'll be going over, and they're all saved as different scripts. First being raw read count processing, normalization, differential expression analysis, functional enrichment, and some examples of data visualizations. First of all, what is RNA-seq and why I use it? So RNA-seq is basically sequencing of RNA molecules, usually mRNA, and the sequencing reads are then aligned to a genome or transcripts, and these reads are counted. Basically, they're counted against a reference of known genes or transcripts, and from this count information, you can do quantitative analysis. So you can use the quantitation to identify genes that have altered abundance under a particular treatment, condition, or disease, and you can use it to look at which RNA molecules are expressed in different cells or tissues. So these are some of the main principles for actually analyzing RNA-seq data. So the first thing that needs to happen is actually pre-processing of the raw sequencing reads, which are in the FASTQ format. And this is not covered by this tutorial. But basically what happens at this step is you take the raw FASTQ files and you process them into files that contain read counts for each transcript. And this is usually done in something like the Galaxy platform or through bash commands. And it's not usually done with R coding, so it won't be covered for today's tutorial. But the next steps will be covered, and they include normalization, which basically is adjusting the read count files, normalizing the count values so you can do proper statistical analysis and data visualizations. The next step usually is differential expression analysis, which allows you to identify transcripts that have altered abundance. And another common analysis that we'll go over is called functional enrichment, which basically lets you identify pathways or groups of related genes that are enriched with the altered transcripts of interests. And this is a good way of helping to interpret what the results actually mean. So kind of the first thing that you'll need to do for the tutorial today is setting up the analysis directory. So everything that you need can be found on the MMID GitHub from this link. And basically all you need to do is you just download everything as a zip file. And when you unzip it, you can save the directory to wherever you want. And then when you open up R or RStudio, don't forget to set the working directory to your analysis directory. And if you do this, you should see something like this in your console, basically just confirming that the working directory has been set properly. So I provided some published data that's publicly available, and this is basically the paper where I got it from. That's looking at transcriptional signatures and preclinical prion infection. I basically chose this data set because it's very similar to the type of RNA-seq data that I'm used to working with, so it just made it easy for me to set up a tutorial with it. And it's basically RNA-seq data from brain tissue from mice that were infected with prion disease. So the idea being to identify any transcripts that are altered in the brains of prion-infected mice. And you should also know that this is a time course study. Gene expression was basically evaluated at multiple time points post-infection. Just something to keep in mind as you're going through the tutorial. And kind of the first thing that we actually start with are these raw read count files. And if you look in the analysis directory, they're saved under the raw read count file folder. And there's multiple files, and each file corresponds to an individual sample. So if you were to open up these files in Notepad or something like that, you can see that it's just a list of genes with a corresponding number. And this number is the number of reads that actually map to that gene. So these are the read counts. And this is the actual pre-processing pipeline that I used to generate these read count files. I feel that it's worth mentioning exactly how this happened, although the point of this tutorial isn't to go over this part of the analysis. And basically the raw reads were cleaned up, they're mapped to a reference genome, and then the reads that were successfully mapped to known transcripts were counted to produce these read count files that are provided for the tutorial. Okay, so the first script is basically all about processing these raw read count files into one single matrix that contains the full data set. So basically, we start out with defining the paths where the read count files are saved using this sys.glob function. So this is just telling R where to actually find all of the files. And then we create an empty list that's eventually going to store each of the read count files. And we use a for loop to basically read each of the read count files one at a time. And they each get added to this list. And once this is done, the list is basically converted to a data frame using the do.call function. And this basically merges all of the read count files into a matrix with each column corresponding to one individual read count file. So the read counts are then cleaned up by removing any reads that had no read counts associated with them, as well as ordering the genes based on the overall average abundance. And then the read count file is saved for further analysis. This is basically what it looks like. It's just a matrix. Each column corresponds to a separate sample, and each row corresponds to a separate gene, with these numbers being the actual read counts associated. Okay, so the next thing that happens in RNA-seq analysis is normalization. And to do this, I like to use the DE-seq2 package. There's other packages out there that you can use for RNA-seq. Another popular one is EnrichR, but we'll go over DE-seq2. So there's two input files that are required to normalize the reads. Basically, we need the raw read count matrix that we just produced in the previous script. We also need a separate file that has sample information. Right, so each of these rows are different samples, and for each of the samples, we can see the time point that the brain tissue was collected at, as well as the treatment. So there's two treatments in this study, basically mock treated animals that were not infected and are serving as controls, as well as the RML treated mice. So RML is basically a particular prion disease that's used in mice to study prion infection. So RML are basically the prion infected animals that we're interested in studying. So this chunk of code here basically shows basic normalization using DE-seq2. So you got to load the input files first. So we load the raw read count file as well as the sample information file. And it's important before making the DE-seq data object to make sure that the actual samples are in order. If you look back here, the sample names are columns in the read count matrix, and they're also rows in the sample info matrix. You want to make sure that they match up with each other. So then you make the DE-seq data object using this DE-seq dataset from matrix function. You specify the read counts, the sample information, and a design formula. So This is basically telling DE-seq how your experiment was designed. So you need to provide the information from the sample info file that you want to test. In this case, the samples are categorized by treatment and time point. And we use the tilde key to specify that this is a design formula. The actual normalization with DE-seq2 happens with this one line of code here. The function is just called DE-seq. It normalizes the data set. And we want to examine the normalization. We can use this plot dispersion estimates function, which makes a plot like this. And it's kind of just illustrating how DE-seq2 actually goes about normalizing the data. This is a plot of dispersion on the y-axis plotted against the mean normalized recount on the x-axis. So dispersion is a measurement of variation in gene expression. So DE-seq2 uses a global normalization method. It actually uses a negative binomial model of dispersion modeled against mean read count. So the fitted model is shown in this red line here. So it's basically the way DE-seq2 expects the data to be distributed. And it basically works by shrinking the read counts towards this red line. And it uses this to adjust all of the read counts towards the expected model. I'm not really a statistician. I can't really go into the full details of how this works, but basically this is how DE-seq2 normalizes, and it's been shown to work. It's basically empirically been shown to work fairly well. And the other popular RNA-seq normalization package, EnrichR, uses a very similar type of model to normalize. So it's a popular way of normalizing RNA-seq data. So once you've normalized your data, you can actually extract some normalized read counts using this VSD function, and you can use it for various downstream analysis. So a good thing to do early on in your analysis is to make a principal component analysis plot, or PCA plot. And DE-seq2 has a built-in function that basically does this using the normalized count data. And I've also shown an example of how to customize the PCA plot using ggplot here. And the final thing that happens in the script is the normalized read counts are saved for visualization later on. But this is what the principal component plot actually looks like. So how this type of analysis works is it's trying to measure the actual variance in gene expression, and it's reducing it down to 2 dimensions, which are called principal components. So the principal component 1 and the x-axis here is taking into account 39% of the variance in the overall data set. And similarly for principal component 2, 18% of the variance is accounted for. And this is just meant to visualize the interrelatedness between samples in 2 dimensions to make it easier to look at. And you can use it to identify sources of variation within your data set. So when you look at this plot, we can see the mock treated samples are circles, whereas the RML Prion infected samples are all triangles, and the different colors represent different time points. So we can see that the RML samples at these 3 time points of the terminal, 20 weeks post-infection, and 18 weeks post-infection, they all cluster very separately from the rest of the data set in the PCA analysis. This is kind of telling us that these samples do have like a strong association with prion disease. So in other words, prion disease is likely to be driving gene expression in these samples. When we look at some of these other samples, we can see some smaller clusters of samples forming, kind of both with the control, the mock samples and the RML samples. These are probably batch effects or biological variation or technical artifacts that might be influencing your data. So trying to do the differential expression analysis with these samples might be less reliable. Well, it's important to do this because it kind of helps interpret how you should set up your differential expression analysis, because you want to avoid any sources of unwanted variation from influencing your results. There's various ways you can deal for it. There are ways you can actually eliminate batch effects, the normalization using DE-seq2, or you can do subgroup analysis by only considering samples within each cluster. That's kind of beyond the scope of what I'll be going over for today's tutorial, but it's something to keep in mind when you're analyzing your own data. So once you've kind of have your normalized data and you've taken your first look at it, the next thing is to actually set up the differential expression analysis using DE-seq2. So this works, it starts out kind of the same as normalizing the data set. You load your read count data and you make a DE-seq data object. But this time what we're actually going to do is we're going to do a separate analysis for each time point. So we'll start out by only taking the samples from a terminal time point. And we're basically doing this by filtering on the sample information data set and pulling out the sample names. So then when we remake our DE-seq data object, it's being made with a reduced data set that only has the samples at the terminal time point. And we are only considering the effective treatment. And then we renormalize the data set for differential expression testing. So keep in mind that based on the PCA plot, this might not necessarily be the best way of actually normalizing the data for differential expression testing, but it's kind of the most straightforward way of doing it for the purposes of the tutorial. It's also the way that was done in the original paper, so we'll stick with it for now. But I just want to emphasize that when you are analyzing your own data, the way you actually go about normalizing for differential expression is something you'll need to think about. But to actually get the differential expression results, we use this results function. We tell it we give it the normalized data set, and we're telling it that we want to compare the RML versus mock samples using this contrast here. And this gives us basically a results file, which I clean up here, and is saved in a folder, the DE results folder for further analysis. And this is basically what the actual differential expression results file looks like. It's just a matrix. Each line, each row is a gene, and we have some statistics for the differential expression testing, including the log2 fold change, which is basically giving us the magnitude of change between the RML prion infected versus mock samples, as well as some statistics like p-values, which give us a measurement of statistical significance. So we also have in script 3 here is a little bit more advanced. So if we want to test every time point for differential expression, what we can do is set up a for loop. That's what I've done here. And it's basically pulling out the samples at each time point and renormalizing the data and getting the differential expression results as before. And it just does this by looping through each of the 8 different time points. And it saves all of the files in that results folder. So after running this loop, what you should see in your results folder is 8 separate files showing the differential expression results for each of the time points. So now that we've done our differential expression analysis, the next step we'll go over is functional enrichment with enrichR. So this is just a way of trying to find pathways or groups of genes that are enriched with your differentially expressed genes, and it's just an easy way of trying to interpret your data. And there's different databases out there that you can use for functional enrichment analysis. I'll be going over enrichR because that's the one I prefer to use. And it has basically an R package that lets you interface with it through R. But there's also a web browser for this enrichR analysis that you can use. Oftentimes, I like to go to the web browser first when I'm doing my enrichment analysis. So that's kind of what's shown here. So we start out by identifying our differentially expressed genes, and we do this by loading the results file. So for example, we're just going to look at the results from the terminal mice. And to get the genes, we basically need to filter the list of differential expression results by various criteria. So these are the criteria that I've usually use. Adjusted p-value less than 0.05, a log2 fold change greater than 0.85 for basically upregulated genes, or less than negative 0.85 for downregulated genes, as well as a base mean read count greater than 15. And I'm basically using deepler here with these pipe commands to pull out the list of genes that actually meet this criteria. And then I'm using this write clipboard function to copy it to your clipboard. So when you do this, you can basically go right to the enrichR website shown here, and you can copy and paste your list of genes. And if you click submit, you'll basically get a pretty big list of functional enrichment results. So this is kind of why I like enrichR. It has this website, and it shows you all of these different databases for functional enrichment, including, and they're categorized into transcription factors, pathways, ontologies, diseases, and cell types. And under each of these tabs, you can see various examples of databases. So under pathways, for example, you can see the enrichment results for Bioplanet, Wikipathways, and KEGG as just a few examples. So we can see that a lot of the enriched pathways have to do with the immune system, cytokine signaling, and microglia, etc. Usually what I'll do is kind of early on in the analysis, I'll take a quick look at the actual enrichR website, and I'll kind of pick out the best databases that actually describe the genes that I'm looking at. Because when you're working with enrichR and R code, you actually need to manually specify a list of databases. So by looking at it on the web browser, I decided that I'm interested in these three databases. So to use enrichR and R code, it has a few advantages. The main one being that you can actually save the result as a file that you can go back to later on. It makes it easy to save your results. So that's kind of the advantage of directly doing it from R. And to do this, all you need is your list of databases as well as your list of genes that meet your criteria. So again, this is the list of genes that were increased at the terminal time point. And then to run it through enrichR, it's pretty straightforward. There's an enrichR function. You feed it these two objects, and it spits out the results. And the results are in the format of a list. You can actually go and look at the results file. So to save it, what I usually like to do is convert it from a list to a data frame. This for loop is just manually specifying the database for each of the entries in the list. And then to make it into a data frame, we use this do.call function. And it's basically creating a data frame that looks like this, where each of the rows are different pathways or gene sets that were enriched. And it gives us some statistics that we can use to rank the pathways. It gives us the number of genes that were present in the pathway and all kinds of information like this. And then what I've also shown in the script is a little bit more advanced. It's another for loop if you just want to compare every possible comparison. So in other words, you want to do the enrichment results for upregulated and downregulated genes at each of the 8 time points. We basically start by making an empty list to save all of our results. And then we loop through each of the 8 time points and repeat that same kind of analysis. So we load our differential expression results, identify genes that meet the differential expression criteria, run it through enrichR, and add everything to the list, which gets converted to a data frame at the end and saved as a single file, which can be used for visualization later on. And this basically brings me to the final thing that we'll be going over, and these are just some examples of common data visualizations for RNA-seq analysis. And the first one is a volcano plot. So this is basically just plotting the p-values against fold change for each of the genes. And to make this kind of a plot, all you really need are the differential expression results files. So here I'm showing an example. We're using the results file from a terminal time point. To make a basic plot, all you really need to do with ggplot is just plot the fold change on the x-axis and negative log 10 p-value on the y-axis. And I've also shown an example of how to make the plot look a little bit prettier by changing some of the settings. And this is basically what it looks like. So yeah, the negative log 10 p-value on the y-axis is showing statistical significance, and the log 2 fold change on the x-axis is showing the magnitude of change. And it lets us look at, the upregulated and downregulated genes, and gives us a sense of the effect size. In this case, the effect size of RML infection at the terminal time point. And for this particular data set, it was kind of a complicated data set because there were 8 different time points. So to make a volcano plot for each of the 8 time points would not really be an effective use of space. So maybe all you want to do is simply plot the number of genes at each of the time points. So I've kind of shown an example of this here. You start by creating an empty data frame that will contain all of the results. And then, kind of as before, we use a for loop, and we loop through each of the results files at each of the 8 time points. And we pull out the number of genes that meet the criteria for upregulated versus and downregulated genes. And yeah, then we can make a basic plot in ggplot by plotting the number of genes against time point. And I've also shown an example of a nicer looking plot that's cleaned up a little bit. And that's basically what's shown here. So we can see that, the terminal time point had by far the largest number of genes that met differential expression criteria. And this makes sense because we would expect the strongest effect size for RML infection at the terminal time point when the mice were suffering the most severe effects of the disease. And so the next data visualization we'll go over is the heat map, which is a very popular one for RNA-seq analysis. So to do this, we're actually just going to make a heat map of the genes that were differentially expressed at the terminal time point. So we do this by just specifying the genes that met the differential abundance criteria from the terminal DE results file. Then we actually use the normalized read counts that were saved all the way back in script 2 in order to make the heat map. So we load these normalized read count files, and we make sure that it's a matrix, and we pull out the genes that we're interested in this, which is specified here. And then we calculate what are called z-scores, which is pretty easy to do. It's just you subtract the mean read count for each entry in your matrix, and then you divide it by the standard deviation. And this is just a popular thing that's used to make heat maps because it makes it easy to look for patterns in the gene expression data. So I use the pHeatmap.R package to make heat maps often. And it's pretty easy to make a basic heat map. You just need to give it your matrix, and it'll produce kind of a generic heat map with default settings. And I've also shown an example of how to make a nicer heat map by changing a lot of the settings in the plot, and it basically ends up looking like this. So yeah, color is basically mapped to z-score, with orange being the genes that have a high abundance and purple being the genes with a low abundance. And these trees are kind of showing clustering of the samples as well as the actual genes, and this makes it easy to visualize patterns in the gene expression data. I've also added an annotation at the top showing the treatment and time point for each of the samples. So we can see that the terminal RML treated samples here have this very different pattern in gene expression. We can basically see the genes that have increased abundance and decreased abundance in these samples. If we go over here, we can even see that some of the samples at 18 weeks and 20 weeks are starting to have increased abundance of some of the genes as well. So this is just a good way of looking at how the data is distributed across all of your samples. And so the final thing that we'll go over for data visualization is actually plotting some of the enrichment results. I mean, technically for a paper, all you really need to show is just a list showing your enrichment results, but there's also lots of different ways for plotting these results. I've basically just kind of shown a kind of a generic way of plotting it. So we start by actually loading the enrichment results file. So remember, this contains like the full enrichment results from upregulated and downregulated genes at each of the 8 time points. So what I usually like to do is just filter out the actual enrichment results that I'm interested in. So we start by filtering out the top 10 enriched wiki pathways that were increased at the terminal time point. So if you do that like this, it's pretty easy to filter out exactly what you want. And we only want the top 10 just to make it easier to visualize. And I like to make a basic enrichment plot by just plotting the negative log 10 p-value against the actual name of the gene set that was enriched. And similarly here, I show like a basic kind of a plot and a slightly nicer looking plot by changing some of the settings. And I'll mention, like, for all of these kind of data visualizations, feel free to try to change some of the settings yourself when you're going through the scripts to see if you can customize the plots on your own. But this is basically what the enrichment plot looks like. Each row is basically one of the pathways, and it's plotted against the negative log 10 p-value, and this is just for ranking, the top 10 pathways. And I've mapped color to combined score, which is a statistic that's produced by EnrichR. And I've even added the number of genes in each pathway as a label. So we can see that many of the top pathways have to do with microglia. This is basically just a type of inflammatory cell that's present within the brain and become activated during prion disease. So it kind of makes sense to see these pathways here. And yeah, that basically summarizes the full RNA-seq analysis, starting from the raw read counts. And the way that I've tried to kind of set up the actual tutorial is there's these 5 different scripts that contain different parts of the analysis. This is because when you're actually doing the analysis, I find it helpful to divide it across different scripts. It makes it easy to keep yourself organized and to go back. If you need to go back to redo a particular part of the analysis, it can make it easier to find what you're looking for. And you also will note that each of these scripts produces some kind of output that's used by scripts in the future. So the raw read count files are used for normalization as well as for differential expression analysis. The differential expression results files are saved and they're used for the functional enrichment. The output of all 3 of these scripts here are used for the data visualizations in script 5. And here are some of the helpful resources that might help you complete the tutorial or adapt some of this code to your own data set, if that's what you're interested in doing in the future. So this is basically the DE-seq2 vignette. It has a pretty good description of how to use DE-seq2 as well as many different aspects of RNA-seq analysis. This is the EnrichR website, which you can use for your functional enrichment. And this is just a paper that goes over some of the main principles for RNA-seq analysis. With that, I'll thank you all for listening and take any questions. Thank you.